Pharmaceutical Processing, in conjunction with Interfex 2017, presents Interfex Live.
Okay. Biosimilars. 3 years out. You believe it, Manny? We've been talking about
I know.
So, we did this 2 years running, this is our third year, and the reason is because of the dynamics involved and the changing environment. We found that every year we're going to revisit it. So, one of the key important things to take a retrospective, and then we're going to go from there. Okay, so we got Manny Krishnan from Pall, and we got Michiel Ultee. Did I get that right? Yes. And we're going to be talking about the specifics on this one, on the biosimilars. But first of all, well, Manny, we're going to be looking to you. Let's do a retrospective from where we were, say, 12 months ago.
Yep.
What's new?
So we've come a long ways. Yeah. So last year when we had this conversation, at least in the US, we did not have any monoclonal antibodies that were approved as biosimilars. And in the last 1 year, we have 2 monoclonal antibodies approved as biosimilars.
Okay.
And that's a huge step for the industry. I mean, that's antibodies, as we all know, are complex molecules, and getting those approved to the FDA as a biosimilar is a huge step for this industry. So that itself speaks volumes in terms of how far we've come since last year.
That's amazing. That's something. So that's one of the poster children we're talking about, right?
Yeah, it's a critical
that's
milestone.
the ones we were dreaming of.
Yeah.
For a while. Okay. So any other noteworthy things that we should be aware of that in Manny's perspective on biosimilars here? You're allowed, Michael. You're allowed.
No, I mean, I would agree. The approval by FDA of 2 antibody drugs, which are multi-chain, post-translational modification, much more complex and larger molecules than previous 2 that they approved, is a big step forward.
Absolutely. So, while we're on the topic, let's talk about it. So what are the properties of protein matter? That if you were going to make a biosimilar, what are you looking to do,
Well, you're looking at the innovator drug in terms of its structure and its functional properties. And you start with all proteins are sequences of amino acids in a defined order and a defined number. To be a biosimilar, you have to exactly the same sequence. You're not allowed to even change 1 amino acid in 1,000. You have to be exactly the same. Now that's not too hard because that's the genetic code to transcribe. But there are other things that happen after the protein is put together called post-translational modifications. The most famous of this is glycosylation, where carbohydrate is added to the protein. And certain host cells can do that, like mammalian cells, like CHO cells, and other more primitive cells, like bacterial, like E. coli, cannot do that. So you need to first see, if you have a glycoprotein, then you need a mammalian cell to produce that. And then you have to see, well, what was used for it by the innovator? Was it CHO-derived? Was it another cell? I mentioned CHO twice because that's the dominant cell line. Chinese hamster ovary is what the 3 letters stand for. That Chinese hamster died a long time ago, but the cells live on for a long time. So, I could go on, but I think you just wanted to know, well, how do you get started?
Yeah, exactly. So in other words, the audience ranges. I mean, we got people, right? But there's also, so where do I start?
So you start with a thorough study of the drug that you want to make a copy of, essentially a biosimilar. And you have to understand that you cannot make an identical copy. That's virtually impossible because of the complexity of the structure and the post-translational modifications, or even if you don't have post-translational modifications, you have differences in slight degradations that can happen to a molecule during production that have really no consequence in most cases to the activity. For example, a glutamine or an asparagine can become a glutamic acid or aspartic acid. That's a process called deamidation. And it's favored in some 3-dimensional arrays of proteins. And so if that's in your active site, then that's a serious issue. If it's somewhere else in the molecule, it probably is not going to make a difference, but it'll introduce a negative charge, and so it'll look different. So any biologic protein is not a purified single substance It's a sort of family of very close relatives, and they all have all the family looks basically the same amino acid sequence, but they got a little few changes here and there, but they're all very close, and that's what you start with, and you have to make your family look like the innovator family, if I want to carry that analogy further. And that's the challenge really in making these drugs, is to try to match them in a number of different analytical areas called critical quality attributes. And those are things like that where you need a charge and where you don't, and so forth. And is your pattern, say, on a charge-based assay, like an isoelectric focusing or a capillary electrophoresis version of that, does it look similar to the innovator, or are your charge variants over here and the other ones are over there? You know, then you're not similar anymore. Whether it acts the same in vivo as a drug, you don't know. But your hurdle to show it's going to be a similar is much harder if your analytics don't match up closely.
Sounds to me like you got to throw a lot of analytical time at this thing, man.
I mean, I think to me, one of the first things you want to do is, get a whole bunch different lots of the innovator molecule and characterize the variability of the innovator molecule, because not every lot is going to look and feel the same. So you want to understand within the innovator molecules spectrum, what are the things that change and how much change is allowed, because they all pass the quality specifications or the innovator company. So you want to basically map out what the variability is for a whole bunch of lots, so you understand what is permissible, what is not permissible. So I think that characterization is a critical component in the process of starting your biosimilar manufacture. Make sense?
Yeah, I would second that, and you should start early, because you're going to have to build up a collection of data based on these different lots of the innovator, because from one lot of innovator drug, or regulatory folks call it the reference product, that product will change a little bit. It actually has a name called drift, and it changes, and so you're trying to see, well, how much drift is allowed and is still in specifications, because you don't know the specifications that the manufacturer got released under. You don't know those, so you have to kind of infer them based on testing a lot of approved lots and say, well, these all got approved, and here's what my results on the reverse phase HPLC look like. And so you need powerful analytics to support this, actually much stronger than you need for an innovator drug, because you're constantly going back to the reference product or doing them side by side and saying, well, and this starts very early on in the cell line development and then through the bioreactor work. And you might choose a cell, ordinarily, a cell line which you choose in an innovator or new drug is the best producing one, that, if you can get 3 grams per liter instead of 2 grams per liter or 1 gram per liter, you're going to go for the higher productivity. Yeah. But in a produced a biosimilar, that one that's cranking out 3 grams per liter might not produce one that's similar enough. So you say, sorry, you don't produce enough, but I'm going to choose another cell. And that's a different strategy than for an innovator drug or a new drug. You're trying to get as much as you can out of your cells.
So Manny, I know one of the questions I've always, and I think it's important to get this, one of the things that I've been involved with, we were gonna make similar not biologics, but we made Me Too products.
Yeah.
And we found that in a lot of cases, accidentally or on purpose, we actually made them better. Mm-hmm. So my question there is, that ain't an option either, because for us it was, unless you want to go back and do a new clinical study, it ain't good.
Yep.
I thought it was a home run, it wasn't.
No, it's not. The biosimilar by definition is similar to the reference molecule. So making it better, either an effector function or half-life in the body, does not make it a biosimilar. So just because you made it better, it's not a biosimilar. You can file as a new molecule, but it's not a biosimilar. So, and that is the reason why I think when we talk about these goalposts, the goalposts are fixed, and you got to hit within those goalposts. And any change outside of that makes it a different molecule altogether. So it makes it harder to meet those specifications.
Yeah. There's no white knight in that particular case. No. So let me ask you this question, all right? One of the things that we found was we go out and we always debated on what end of the batches we should look for, right? So for example, we try to get 10 batches, we try to get 5 batches, You know, it's a tough one, right?
Yeah.
You can wind up buying out every pharmacy within the tri-state area. Secondly is, how far removed from it, and I think the guys in biotech have a unique problem, because when I'm looking at it from the delivery standpoint.
Yeah.
You guys got to look at it from fill finish and work backwards
Yeah.
back into the process. So I think you got something unique there. How do you work around that? So let's talk about the end first, and then let's talk about how do you say, okay, I got the dosage form, but does that really give me a window on what I got to be making? Is that a reasonable question?
Well, your active ingredient, your API, as you call it in small molecule, or bulk drug substance for the biologic, is really not supposed to be affected by going into the vial and adding some excipients. Excipients are supposed to be inert, non-active ingredients, so you know what the active ingredient is, but even if you are limited to the final dosage product. And these products are all injectable. There are no tablets or pills because the proteins can't survive going through the usual GI route. So they're all injectable liquids or freeze-dried products. So in terms of working backwards, yeah, that is something you have to live with in doing this kind of work. But you can learn a lot from the protein qualities. The excipients that are added are typically small molecules that can be quickly removed as part before you begin your biologics testing.
I mean, I think to go back to your the question, to me, I believe at least because of the variability that's involved, and you're trying to map out the variability. More lots you have, the better is your visibility to how much variability is tolerable, right? If you have three lots and the 3 lots don't show very much variability, then you've set yourself a very, very narrow specification, which is not necessarily to your benefit. So you want to get as much variability as possible. So to me, 10 to 20 lots is not an unreasonable number if you can get it. I think that's where again, that gives you a better visibility for how much variability is in the reference molecule. And then you work off of that and try to understand how you should design your critical quality attributes in order to hit those goalposts.
Yeah. And it's also known that the product produced in one part of the world might have a little different properties than a different other part of the world.
Yeah.
So you have to look at where you're going to market this and focus on the area there. Now, if you're going to do global, then getting all those areas,
Absolutely.
collecting samples not just in the U.S. but in Europe and other countries as well. It's worthwhile.
Absolutely. And the other thing also is, certainly the active molecule is critical, but you also want to make sure that there are no other formulation ingredients that render activity to the drug, right? So you want to make sure if there are other ingredients in the formulation that are also active other than the protein of interest, you want to understand that as well. The rest of the excipients don't really matter, but the ones you want to make sure that you understand all the ones that render some activity to the final drug.
So if I was an innovator company, and I know you're looking at, so all of a sudden they see an uptake in the sample sales. So what's the reaction? There was a point when we ran some major league clinical stuff in cardiovascular to the point where we used to get calls from other companies simply, why is all this stuff disappearing, So my question is, what are the innovators thinking about all this? Before we get into the science, I just want to make sure I understand that.
Well, I think they're going to make it as difficult as possible.
Yeah.
Really,
To get lost. Even though it represents some lost sales, it's a small fraction of their market. Generally you don't go after a drug as a biosimilar unless it's a substantial product, something with billion-dollar sales or something. You're not going to go after the ones that are small, where somebody who buys a lot of these samples for analytics, that might be noticed. But still, I think they're going to make it restrictive, and I haven't actually gone through this process, but I understand you sometimes have to take extraordinary means working in different countries and so forth.
I just asked that because I noticed the legal community seems to be getting very happy with this whole situation.
Yeah.
So let's. So let me ask you this. It's one of the things we've been talking about on biologics. We've been talking about, continuous process intensification. I don't want to get involved with all of that, but I think one of the things that I think the environment people are looking at now is It's not we're not entering into traditional situation. They're either looking at disposable situations, single-use situations. Does that change anything from the biosimilars? I mean, if we're trying to get the right fingerprint, I mean, do we have to make it an identical environment? You know what I'm saying? I mean, that's a real challenge. So what's your take on that?
It's a very good question, right? So, to me, it's critical to understand what are the CQAs, the critical quality attributes, right? Once you understand what the critical quality attributes are, and as Mike talked about earlier, as long as you're making in the same cell line, the rest of the process should not really matter. So whether you run it in a single-use facility, single-use processing systems, continuous or batch processes, it should not really make that much of a difference. The advantage that single-use brings and potentially continuous brings to the table is the fact that with a lot of these biosimilar molecules, the market acceptance is not fully clear. So we do not know what the addressable market is, what the available market is, because we do not know how market will accept it. Because of the market uncertainty, you want to make sure that you set yourself up with a facility that has the flexibility, right? So that has the flexibility to make as many batches as you need or as few batches as you need, and you have the flexibility to reduce your capital investment to as little as you need to make a GMP product, so that if you don't have sufficient market uptake, at least you're not burning a hole through your pockets. Right? So that is where single-use technologies and potentially continuous processing technologies actually have a role to play. Because you can significantly defer when you make the investment. That's very critical. So you don't need 5 years or 4 years to build your facility and qualify the facility. You can do it in much shorter time. So you have the flexibility to defer the investment, and also you have the advantage of having a smaller investment and a smaller facility that gives you the flexibility. So that is where with biosimilars, because of the uncertainty in the marketplace, this flexibility becomes important. Right? So single-use and continuous processing certainly aid that process.
Yeah. I think what Manny is describing very well is that it lowers the bar to entry. So it's the high investment cost of building a stainless facility and the associated utilities and so forth is a significant barrier. But if you can reduce those by going to single-use solutions, then you can get in easier, and you can also change over to other products.
Exactly.
Much easier go towards making a multi-product facility because any of your product contact materials are tossed out.
I think the other thing too, and I think a lot of the process owners look at, and at least in in a lot of the conversations we've had, is that I think if you're looking to do something that, and I don't want to get too far off the topic, but if you're looking to optimize continuous, the answer is you should pick within the process chain what needs to be optimized. In other words, maybe it's upstream is where you need it. You don't need it in chromatography, right? I mean, that's the key. I mean, I think people always think the totality. Yeah. I think you pick and choose. Would you agree with that, Mike? I mean, to me, that would seem to be, you got to be judicious of what you say I'm going to use. What do you think about that?
I mean, upstream, I think you have a choice of a continuous process, be a perfusion process. Historically, that brought some headaches, but it was used when you had to because the molecules are, that are more delicate can be pulled out of the bioreactor sooner. And that's still, I think, the primary rationale to go that way. But on the other hand, it does give you an opening toward continuous processing and going right from your upstream continuously into downstream, rather than batching all the upstream together and then going forward. Downstream, the reason I think for continuous processing is more some savings of there's higher capacity, say if you have a simulated moving bed type system, then you can fill each smaller column to its capacity, and instead of going to a dynamic capacity, you have what's called static capacity on each chromatography column. And people are taking shorter steps, let's just say combining two unit operations in one. If you can get conditions, say, on a cation anion exchanger that are similar, then you can flow from one to the other. And there you save a unit op, essentially. So you're sort of kind of pushing steps together and sort of approaching a continuous downstream process. And it's possible with specialized operations to do that, like the BioSMB and so forth. I know that Pall even has a lab to demonstrate this technology on a small scale. So it's possible to do that. Whether it's the big reason to do it in terms of biosimilars is try to keep your cost of production down.
Yeah. I mean,
Because costs become very important as you get into a competitive market.
Absolutely. I think the other thing where some of the continuous technologies also help is the discussion we had earlier about the market uncertainty and how much do you have to produce, right? For example, I mean, if you look at the various unit operations, the most expensive unit operation in a downstream purification step is your protein A. And if you're only going to be making 5 batches a year, and each batch takes 5 cycles of protein A, you have a very large capital investment of protein A, because you can use protein A for 200 cycles. Now you're only making 25 cycles of use per year, and you can't keep it for too many years either. So now you essentially are using maybe 1/3, 1/4, or 1/5 of the protein A capacity and throwing away a whole bunch of investment. And this is where continuous can actually help, because now you're cycling the columns more often. You're getting the benefit of higher capacity, but also you're getting the end of life much faster. So you actually can get to an end of life in 1 year in 5 batches, because you're cycling the columns much more often. So there are other intangible benefits of going down this path, particularly when there's market uncertainty. When you know how much you have to make, you know exactly what the demand is, and you know the demand profile, then you can certainly plan it appropriately. When the uncertainty is there, you need to build flexibility, and that's where some of these technologies help you build the flexibility.
So let me ask you this then. Sounds to me like your target molecule, complexity, non-complexity, getting back to what I said before, where do you start? Like you said, you spend the time on the analytics, but how complex that is drives this. It sounds to me like that's going to be the driver, not only, right?
Well, sure. If your molecule has no glycosylation or other post-transition modification, your job is easier.
Yeah.
And your choice of expression system is wider. If you have a more standard glycoprotein, something like a monoclonal antibody or some larger enzyme, then your choice of expression system is more defined. You really should try to first start with what the reference product is made. So if the reference product is made in a particular mammalian cell line, like a CHO or N0, I would start with that, because there's subtle differences between even those, they're both mammalian cells, but they have slightly different glycosylation profiles. And so, CHO is the market leader. 75% or more of glycoproteins are made in it. But some of these earlier drugs were made in some other cell lines. And you may be able to reproduce it in CHO. If you can, then you show through strong analytics that you're producing the same structure. And so you can do that. But if you don't have the PTMs or post-transition modifications, then technically you could make it in any system that can produce a protein, even bacterial.
So let me ask you this. So it sounds to me like, let's get back to where we were on CQA identification. So let's talk about how do you identify that so you got those. In other words, how do you determine the acceptable levels? Sounds to me like complexity is number one, right? That's got to play as a factor. So why don't we walk through that a little, okay?
Well, you need, charge determining assays, because charge can make a significant effect on not just the binding of the molecule to its target or its activity, but also its pharmacokinetic and pharmacodynamic profile, how quickly it's eliminated from the bloodstream, which can affect your dose. And to be a biosimilar, you have to have the same dose. You can't, as I think you alluded to earlier, Russ, suddenly you find out yours is 10 times stronger and you can use a low dose. Well, that's what you call a bio-better, but that's a different route. It's no longer biosimilar, and some companies are making bio-betters because they think that's more sensible than making a copy of an old drug. That's a business decision. But in terms of back to biosimilars, you're trying to match that innovator or comparator product. And there's something exercise that some of you may be familiar with called a comparability exercise. When you make a change in manufacturing, you have to show that your new product produced after that change looks comparable. Well, this is kind of similar. You're doing a comparability exercise every step of the way in building a biosimilar. And in building the cell line, say you decide you're going to produce it in a CHO cell. Well, then it's choosing the clone, because from one clone of cells to the next, you can make different types of structures. And not just the amount that you produce, but also some of the critical quality attributes like the difference. So I mentioned the charge, and the other thing is, the binding for the target, that should be the same. There's some other parameters that you're looking for. Classically, you're looking at the extent of aggregate. Usually the lower the better. You're trying to match up. And if your material produces less aggregate than the innovator drug, that may be okay, because that's generally an undesirable one. And if your process produces something with fewer impurities, such as host cell proteins, those are essentially inactive products anyway. So you won't be challenged on your biosimilar if you have fewer impurities in your batch. So you shouldn't try to deliberately make your process less pure. That's not the intent of any of the regulatory agencies.
Well, I mean, there's clinical downsides to that too.
Exactly.
Yeah, and those can cause allergic effects. And the other concern about aggregates are that you can produce higher levels of immunogenicity or the immune response of the patient. So you want to keep those minimal as well. But there's a series of characterization studies that you do in developing any new protein drug. And you have to apply those earlier, which is using highly sophisticated mass spectrometry, for example, LC-MS, MS-MS, so forth, to really characterize the structure that you have something comparable to what you have on the market.
Yeah, go ahead.
I have a question for Mike. Is it okay if I ask him a question?
Yeah, as long as it's not hard. Yeah, okay.
So Mike, there are molecules, reference molecules today that are produced and administered as IV, right? So if a biosimilar manufacturer wants to make the same molecule but administer as subcutaneous, how would the regulators look at it? Is that considered a biobetter? Is that a biosimilar? How is that viewed by the regulatory agencies?
Well, my understanding is currently the biosimilar is to have the same route of administration. However, I think the FDA would encourage this kind of change because it's much more convenient for the patient. So you can have at-home treatment with subcutaneous delivery, just sort of injected under the skin, as opposed to IV, where you typically need to go at least to a doctor's office or a clinic. So you need a medical professional to administer IV. And that generally reduces patient compliance, and it adds to the cost and reduces the availability. So you can do at-home delivery subcutaneous. So I think the regulatory authorities would work with you towards getting that route, as they have done with other companies who have innovator drugs are moving to a second generation that's going to be, say, fewer injections by a subcutaneous injection of a highly concentrated small volume that slowly dissipates over time. So, but it's an interesting question whether, currently, as I understand the guidelines, you must have the same route of administration, but I would have that discussion with the regulatory folks, because I think they want to encourage availability of these very complex and expensive drugs.
Well, I had another question before too. Is that, and I don't mean to keep wrapping this around, but I think it becomes a strategy question. And that was, we were talking about continuous bioprocess. Somebody in the audience said, well, there's like 20 continuous processes approved. So I thought that was pretty good, As it turns out, they're legacy products.
Yep.
All right? My question is this. So I'm going to take a run at this winter drug, right? And my guess would be batch process. You got to do it. In words, if you went in and you went semi-continuous or whatever, you got to stick to the script, or can you go in with a continuous, or how much flexibility up and downstream do you got? I never thought about that one,
Well, I haven't seen that in the projects I've looked at, but to go
fair enough.
Switch off, I think you could make the argument if you measure there's something people who build biosimilars are aware of this, something called acidic variance, and that means that deamidation I spoke of, or there's extra sialic acid to add acidic groups to it, or a loss of positive charges. There's a C-terminal lysine on many IgGs, and it comes off easily. So you lost a plus charge, it's kind of the same as adding a minus charge. And so these acidic variants you need to have it comparable looking to the innovator. And so a concern that you might not get there by going with, say, a continuous perfusion process, you might reduce that level and you might not match up. Whether that's important to but it's a critical quality attribute,
Exactly.
requires some extensive testing. But you'll probably have to do more testing than if you match it up. So,
I mean, that's the stuff that's behind the curtain.
yeah.
That in other words, you can win with your biosimilar. That's a card they can play later on. I mean, it could be a downside. That's all
You'd
I'm-
already know that because
Yeah.
you look at your profile and you have samples of the reference product,
Exactly.
and a number of them, and you say, well, here's my goalpost, my
Yeah.
playing field, and I'm right in there, or no, I'm not. I'm halfway out of it, and so you have to say, well, but wait a minute, I had a clone here that could produce it right in the middle, even in a continuous process. So that clone may not be the winner clone in productivity, but it might be the winner in terms of comparability to the innovator product.
I think to add to Mike's response, I think from a downstream, from upstream, I can see potentially, how represented the molecule is with the reference product. From a downstream purification technology standpoint, as long as you're able to meet the CQAs of the reference drug, I don't think it really matters if you produce in a semi-continuous, continuous, or a batch mode. The critical thing is, are you able to meet the critical quality attributes? And most of the time, the biosimilar manufacturers don't know exactly how the innovator drug is manufactured in the first place. There are a lot of nuances in terms of how the elutions are carried out, what the buffer chemistries are used, even the wash cycles and chromatography columns. You do not know all the details. Right? So to say that just because you run a batch process, you're going to produce an identical molecule is a fallacy. So at the end of the day, are you able to meet the critical quality attributes or not? If you're able to meet the critical quality attributes, it doesn't matter if your sequence of columns is backwards. So that's the reason, to me, it's dangerous to get fixated on a batch versus continuous and ascribe a difference just because it is a continuous or a batch. It has to be different. I don't think that's necessarily the case. It is, are you able to meet the critical quality attributes of the reference molecule? If you are, then it's a biosimilar.
Yeah.
That's the way I would look at it. And I think the regulators are pushing for that kind of determination rather than get fixated on the process. Get more into in understanding what are the right quality attributes you want to focus on, and then focus on them, and the process needs to be designed to be able to deliver those things. And that's true for innovator molecules, it's true for biosimilars.
And you also need to focus on the mechanism of action, or MOA,
Right.
of the innovator product. So if your mechanism of action is an antibody and it just blocks a target and shuts it down, okay, your focus is going to be on the binding site of the antibody. But if it also includes what's called effector function, which is on the tail of the antibody, where a particular group of enzymes called complement comes in and recognizes that bound antibody and starts attacking the cell it's bound to, that, say, may be important in say, an antibody against cancer, like Herceptin. And in order to generate a desired immune response, you don't just want to block the receptor, you want to recruit the immune system in there through the complement. It's called antibody-dependent cellular cytotoxicity, or ADCC. And that depends on the structures in the FC, and specifically the glycosylation patterns. So you can, if you have, for example, what's called a branching fructose from a particular kind of sugar, then your ability to interact with complements reduced. And if you eliminate that, you have a threefold higher increase. So you're looking, you might make, again, like I said, Russ, a bio better, but that's not what you're trying to do.
Exactly.
Here, unless you want to change horses in midstream and go after that. But, you know, and finally about bio betters, it's not as hard as doing a new molecule from scratch, because you know your patient indications, you know a lot about the basic molecule, you've just made it better. So it's sort of between a brand new molecule and a biosimilar.
One of the things that I think we were talking about in an earlier session too was enablers. So you understand, one of the things are small cap companies trying to get into the pharma market, and the conduit. We were talking about reasonable scale equipment, the utilization of CMOs. Who's the enablers here? I mean, what role are just on the biosimilar? Are the CMOs or go-to enablers, or where are we at here? Is it two different markets? Is that a bad question?
Not at all. It's a great question.
All right. I want to make sure.
Yeah, go ahead.
Well, the CMOs will get you, they have a lot of opportunity to work on different proteins, and they have a lot of knowledge base on similar molecules to yours if they've been in business for a little while. So there's a lot of advantages to working with that. And if you change your mind, you say, well, there's 3 other people already in this space and I'm going to drop out of it, then you haven't invested as much as you have if you built your own facility. So there's certain advantages. Now, if you already have a facility or a larger company and you're saying, well, I've already got this, I'll just use the CMOs as I need to supplement what I have. I think the CMOs would be an enabler because they have the expertise already in-house on a sort of multi-product, wide variety of products, and chances are they've made a product similar to the one that you've already done.
I think the critical thing, I'm sure Michael agree, when you're manufacturing a biosimilar, you want to go to a CMO that has very, very strong analytical capabilities. Right, because one of the critical things you're trying to do is match the reference molecule. So the criticality of the analytical capabilities becomes much, much higher for a biosimilar compared to an innovative product. So,
yeah, that's certainly true. And certain CMOs or CDMOs, as many prefer to be called because they do development, not just manufacturing, varies in terms of their biggest strengths. And some are really strong analytics, and but maybe their production, they have a limited set of bioreactors. Others produce a lot of material, but their analytical department might be weak. So you want one, as Manny says, has a good expertise, a really strong expertise in analytics. And can't stress enough that that's a dominant trait you need to look for in any production facility.
And I think there's got to be some. One of the things we do on a regular basis too is we talk about who owns the IP.
Yeah. I
think the CMO's it's kind of between a rock and a hard place in those situations.
Yeah.
Not to mix metaphors, but okay. Thank you very much.
Thank you.